It's my pleasure to be the moderator for the 10 p.m. session here. This session is devoted to two very excellent people in the field of spatial proteomics and analysis tools. Stellar is a tool designed and published by both John Hickey and Jure Leskovec. And in that, they can show you how to determine the cellular neighborhoods in multiplexed antibody-based imaging technologies. Nature Methods actually had named spatial proteomics as their method of the year, published in the December 6th issue of Nature Methods this year. So quite timely and very excellent. And without further ado, I'd like to introduce John Hickey, who is an assistant professor at Duke University in the bioengineering and bioinformatics and statistics department. He's also a member of the Center of Advanced Genomics and Technologies and Biomolecular and Tissue Engineering. And without further ado, I'd like to introduce John Hickey. I don't see him though on. Meryl, is he on yet? I don't see him.
No, John hasn't joined yet.
Okay.
You might like to run Jure's video then?
Jure Leskovec could not join us in person, but he provided a video of his talk.
My name is Jure Leskovec, and today I will be talking to you about building foundation models for the AI virtual cell. Cell is fundamental unit of life, and whether you want to understand a healthy organism or you want to understand the disease, you want to cure disease, it is understanding to understand and being model cells. Recently, there have been amazing advancements in the field of AI. At the same time, in biology, we are generating new and exciting data at the scales never possible before. So this allows us to truly rethink how are we building models of biology, how are we building predictive models, machine learning models that allow us to make inferences from the data we are collecting. And what I will be talking to you about today is our perspective that we just published in Cell called How to Build a Virtual Cell with Artificial Intelligence. And I'll show you how we can do this and some early work we've done in this direction. So the goal is to simulate biology. And simulating and predicting biology is very, very hard. These models need to be multiscale. There is a lot of nonlinearity, nonlinear dynamics, the amazing amount of stochasticity and noise. The cell is being measured, interrogated in different ways, and many times the measurement technologies only give us a very incomplete, very partial view of what is going on in the cell. So the goal of this is to create an interconnected framework of foundation models that would allow us to simulate increasingly more complex and more dynamic biological systems. And the key idea to do this is that we need to model biology across scales. We need to model biology at the molecular scale, at the level of DNA, RNA, and for example, proteins. We then need to take these molecular scale measurements and put them into another kind of at the next scale, which is at the scale of a cell, where we focus on modeling a single cell. And then, of course, cells group together in collections, they group into tissues, and understanding spatial relationships is very important to be able to model, let's say, multicellular scale organisms. And if we want to build models across these three scales, we can interconnect them into what we call an AI virtual cell, where basically we have the molecular scale foundation models that can then feed the information to the cellular scale foundation model. And then a collection of cells can be then fed to the next level model with all the descriptions of individual cells that builds us then the representation of the collection of the cells. So the key idea here is that we want to be building what we call universal representation spaces because they unlock fundamentally new capabilities in biomedicine. The idea is that we want to have one single universal information theoretic representation of the cell. We want to have one embedding of the cell. And this cell can be in any kind of state, can come from any organism. We should be able to create an embedding of the cell regardless of the experiment, remove the batch effects and things like that. And if we are able to take these cells and map them into one common space, then we can start comparing them. We can build reference atlases. We can start tracking how the cell state is moving through this space. We can start predicting how it's moving. We can start modeling intrinsic as well as extrinsic perturbations of the cells. We can start thinking about digital twins, spatial niches, in silico experimentation, and so on. So the idea is that this type of representation that is universal has to appear at all three scales, at the scale of molecules, cells, as well as a kind of multicellular systems. Out of this representation, also the idea of having virtual instruments is born, because it's not only enough to capture the information of the cell, but we need to be able to predict what are the future states of the cell, or we need to be able to read out from the representation something that is relevant to us, which is what we would call decoding. So we define two types of virtual instruments, and what we call virtual instruments that can manipulate embeddings, where they take a cell and they predict what is the future state of the cell, or they take a cell and the perturbation and predict what is the post-perturbation state of the cell. And we can also have decoding virtual instruments that take an embedding and do a readout. For example, this could be like a reconstructing from the state of the cell, predicting the phenotype from the embedding of the cell, or type of the cell from its embedding, and things like that. So today I'm going to talk about this vision of the AI virtual cell across all these three different scales, and I'm going to give you some vignettes and some of the first steps we've taken to actually demonstrate viability of this approach. So first, I'm going to talk about the molecular scale embeddings, and just to mention them quickly, here we are thinking about encoding the kind of the molecules of the central dogma of biology, so DNA, RNA, and proteins. And one example, there are many examples at all these different modalities or different types of molecules, but the one prominent example are the protein language models that encode the entire protein universe. They are motivated by AlphaFold and ChatGPT, trained over 250 million plus proteins, and they basically can take a protein and give us a protein embedding. And these embeddings capture both structure, molecular properties, as well as kind of orthology information. Now that these models like ESM have been built, the question is, how do we go to the next scale? How do we take the molecular scale embeddings and bring them to the cellular scale? And here is where our work starts with a paper and approach that we call universal cell embeddings. So the idea of universal cell embeddings is that we want to take advantage of all these cell atlas data sets like cell by gene and other collection of cells where we have different cells coming from different organisms. And the problem today is that different experiments kind of have different types of biases, so we cannot embed all these cells into the same space. And the second problem is that organisms don't have the same genes, so we cannot take easily organisms with different genes and embed them into the same space. We have to do kind of alignment and things like that, and that creates all kinds of problems. So our goal is to create one universal space into which we could embed any cell coming from any organism, and these embeddings would be informative. They would capture biological, they would kind of capture the biology. So here I have a mouse and a fly and a human all embedded together in different colors. You see how kind of cell types nicely align and things like that. So the idea is that these universal spaces are fixed. So this means that these models are pre-trained and then they have the zero-shot capability, which means we can use them without fine-tuning, without any data labeling. So when we get the new data coming from the new experiment, and we want to kind of compare it to the existing data, today, what you have to do is you have to take the new data, you have to label it, you have to build a new model, and then you train the model to be able to kind of see how does your data compare to what has already been known out there. With our types of models, no retraining is needed. You get the new experimental data, you feed it through the model, and you can directly map it into this universal space and read out whatever you would like to learn or read out from the data you have. So we build a cellular scale foundation model that takes the information from the molecular level foundation models and uses that to build now a better representation of a cell. We call the model the universal cell embedding model because it is able to embed any cell from any experiment, any tissue, any organism into the same universal space. The key modeling choices that we made in building this model is that gene expression is not natural language, so we have to develop various kinds of innovations to kind of have inductive biases that is different than the natural language bias. The universal space is fixed, so our model has what is called a zero-shot ability. It can be used on new data, on never seen tissues, never seen organisms without any fine-tuning or any retraining required. And it's trained in a fully self-supervised way, which means the organization that the model learns of the embedding space is fully emergent. There is no data labeling required. The model really just learns from the biology itself. So the way this works is that we basically take the single cell RNA-seq data that includes the gene expression levels, but then for every gene, we then look at the protein that the gene codes for, and we are using the embedding of that protein, so the molecular scale representation, as a way to describe what the gene is, and then send basically now the sequence of genes as encoded by their corresponding protein embeddings into the large transformer architecture to then be able to predict the gene expression level of other genes that the model has not seen. And we trained this model with 33, basically it's a transformer architecture, 33 layers, 650 million parameters on 24 A100 GPUs for about 3 weeks. And then we looked at what the model said. And it was truly remarkable because we were able to take many different cells from many different organisms, many different tissues, experimental conditions, donors, and so on, and the cells and organisms that the model has never seen, and we were able to embed them into the same space. And here you see on the left how the structure of the space with the different cell types now color-coded on top with different data sets, different tissues, as well as different species. And what you notice is that these colors are tight, but the clusters are very tight, nicely integrated with each other. It's not that, one species would be in one part of the space, but the rest of the species would be in the other part of the space. Everything is kind of nicely mixed because different species can have similar or same cell types. That's the interesting thing here. The second thing that is quite amazing with this is that we can take organisms that the model has never seen during training, and we can embed them into the space, and basically the structure of cell types is nicely preserved, nicely emerges from this embedding space. So no blast, no alignment is required. Just send in the proteins, send in the genes and the proteins they code for. What I show you here is 4 different organisms that the model has never seen: a macaque monkey, a mole rat, and 2 data sets from the chicken. The model embeds all these cells, not knowing anything about them. If you look the structure of the embedding space, we see the emergence of this clusters. Here, actually, we color code every cell with the cell type that was externally defined by human labelers. And what you notice is that basically the cell type structure is represented in this embedding space without the model ever even knowing what a cell type is. The model is just learned on basically the gene expression data. No other external labels are necessary. So we see this very interesting ability. The second thing, when we investigate the properties of the embedding space and the distances between different cells of in the embedding space, we see that the structure of the embedding space and the distances very nicely mimic the relationships of different cell types and how they relate to each other. And that's quite interesting, again, because the model has never seen a definition of a cell type. It is trained just purely on the gene expression data, but basically the structure of cell types very nicely emerges, and the model is able to kind of, the embedding space captures kind of the known biology. It naturally emerges. So that is the cellular level foundation model. And now I can give you an example of a virtual instrument that we can build on top of this cellular level representation. And the method is called GEARS, and it's for predicting combinatorial perturbational outcomes from pairwise perturbations of cells. And the original paper was actually developed without the use of universal cell embeddings, but now in the new versions of the software, we have added also the cell embeddings, and that greatly improves the performance of this model. So the problem that we want to solve is we want to predict the outcome of genetic perturbations, right? We have a base gene expression level as an input on the left. Somebody selects what genes they want to perturb. Here we are interested in perturbing pairs of genes. And then on the output, we would like to predict the post-perturbational state, so the gene expression levels of the genes after the perturbation. And this is a fundamental problem in biology. And of course, doing this computationally is very important, because the perturbation space is just too big to be able to be explored experimentally, to be explored manually, especially when you go to pairwise perturbations, triple-wise perturbations, and so on. The space is just too big to be able to explore manually with and experimentally. So we need to kind of explore it computationally. If we are able to do that, there's a lot of really cool applications that we can build on top of this. This can be used for drug target discovery, can be used to identify genetic interactions, can be used for cell engineering, and so on, right? Could really help us speed these different important applications. So our approach is called GEARS. And as I said, on the input, the approach gets the base gene expression levels of unperturbed cells, as well as the indication of what genes we would like to perturb. And then through the neural network and use of knowledge graphs, it produces on the output a gene expression vector of the perturbed cell. So the input is a 20-dimensional vector plus IDs of two genes that we are perturbing, and the output is another 20,000 dimensional vector with a predicted gene expression level of every gene. And when we look at the results of this model, we see that it's able to model and predict non-additive and kind of highly non-linear genetic interactions. For example, here I'm showing pairwise perturbations, where we are looking at the total effect of the perturbation. This is the gray bar. We are then seeing the prediction of GEARS. This is the red bar. And what the yellow bar is, if you would perturb the first gene individually, you would then perturb the second gene individually and just add up the effects, kind of stack up the two effects. And you can see how the GEARS is more accurate, much more accurate than this simple linear model. And the second thing this also demonstrates is that multiple gene perturbations give you highly nonlinear results, right? In some cases, the expression level is actually much bigger than the individual. In other cases, it is actually smaller than individually perturbing the genes. Overall, we find that GEARS has about 2x accuracy of the strongest other baselines. An amazing ability of GEARS is that it can generalize its predictions even to genes it has never seen before. So what we are showing here is, for example, on the left, during training, the model has never seen any cell that would have either of the two genes that we will be perturbing at the test time seen during training. And you see that, of course, kind of the performance from left to right increases because the model sees more information. But on the left, the model has never seen a perturbation of that particular gene, but it can still predict it relatively well. On the right, we see the result of the experiment where the model has seen individually the first gene perturbed, individually the second gene perturbed, never seen a combination of gene perturbs, the combination perturbed, but it can still predict the outcome of the combinatorial perturbation quite accurately. And of course, this was kind of an example of a virtual instrument that allows us to give predictions of genetic perturbations on single cells. But now we are going to move to the multicellular scale, where we want to be able to model kind of tissues that are organized as a spatially organized collection of cells. And the paper I'm going to mention here is a method called Stellar that allows us to do cell type annotation of spatially resolved single cell data. So what are we looking here is we are looking at how different cell types are organized in a tissue. And we want to capture spatial context of cells and understand how different cell types kind of organize in the three-dimensional space. And there are these spatial single cell methods that allow us to collect both the three-dimensional positions of the cell, as well as some subset of genes or gene expressions, not the entire 20,000 dimensional gene expression vector, but some smaller, lower number of genes can be measured. And of course, what we'd like to understand is how these tissues form, how healthy tissues differ from diseased tissues, and so on. And the key approach here is the use of graph neural networks. The idea is that we want to basically take these cells that they appear in space and create a network of interactions between cells based on their spatial proximity. And now that we have a graph of these cells interlinked and kind of interacting with each other, we can now build neural networks that basically learn from this graphical representation. That's the first innovation that we need to make this work. And the second innovation is what we call uncertainty-based adaptive max margin sampling that allow us kind of to learn how to learn at equal speed in all parts of the space across all different cells. This is important because certain cell types are very easy to learn and certain others are very hard to learn. And if you're not kind of learning at equal speed across the entire thing, then you can just kind of focus on one class too much, and the model does not perform well. So our method is called Stellar. It takes as an input unannotated spatially resolved data set. It also has the annotated reference spatial data set on which the annotated reference cellular graph is first built. And then the method takes that annotated reference cell graph, takes the unannotated target cell graph, and then through the use of graph neural network, it assigns cells in the unannotated data set to cell types in the reference data set, or actually tell us that there are novel cell types that don't appear in the annotated reference data set. So the cool thing here is that we don't only try to transfer the cell type labels, but we are also able to discover which new types of cells appear in the unannotated target cell data set that may not appear in the reference data set. When we tested this out, we've seen really good improvement in terms of accuracy, in terms of assigning cell types based on when comparing it to expert-based annotations when compared to prior methods. And we also see that the use of spatial information actually helps us resolve cell types much better than if we would just focus on the molecular information alone and would not consider the three-dimensional structure of cells. So as a case study, we ask ourselves, can we annotate cancer donor tissues using healthy donor tissues, right? So we'll use a healthy donor tissue as a reference. We will get an unannotated cancer donor tissue. And the question is, can we kind of annotate the unannotated tissue from a cancer patient? Can we identify the cell types that are shared, and can we discover the new cell types that do not exist in the healthy tissue? So just to give an idea, these are I'm showing you the ground truth labels obtained through Codex multiplexed imaging data. Every dot is a cell, and every color represents a cell type. This is for the reference for a healthy human. And here you see on the right hand side, you see the unannotated, unlabeled tissue. And you see that it has very different cell types and very, very different structure. So the question is, can we learn on the healthy tissue and kind of make accurate predictions on the diseased one? And there are 12 different cell types in the healthy tissue. So what we find that when we compare the expert annotated ground truth with the machine learning predictions, we can see very good correspondence. Here you see basically the same colors. And what we also see is that the method is actually able to identify the novel cell type that was actually not part of the labeled healthy reference tissues. So the new cell types that appear in the disease state, the method was able to identify those accurately as well. So this is kind of what I presented for you today. Is really I wanted to talk about this idea of using AI to build a virtual cell, to basically build a collection of foundation models that can be interlinked together to create representations, embeddings at the molecular scale, at the cellular scale, as well as at the multicellular scale. And then we have, now that we can create these embeddings, these fixed universal representation spaces, we can build applications, we can build virtual instruments that operate on top of this embedding space. And I showed you, for example, how we can use these virtual instruments to create reference atlases with my mega cell atlas. I talked about gears that can model intrinsic perturbations in terms of genetic perturbations. I talked about spatial organization of tissues and how cellular representations can be used to be able to make accurate inference. And the beauty here is, in this kind of multi-scale approach, is that every kind of higher level scale is getting all the information from the lower level scale. And I think this is kind of an exciting research area and an exciting way to go forward. I will just flash here are all the papers I was mentioning, if you are interested to read that, and would just like to acknowledge my group and all our collaborators, as well as funding partners. Thank you so much, and thank you again.
All right, I would like to now introduce John Hickey from Duke University. He will be talking about cellular neighborhoods.
Thanks for having me. This is really exciting to be here, and an honor to follow Jure and his work, the amazing work that he's doing. So today, I'm going to talk, like Ellen said, about neighborhoods. But first, I wanted to start with a question, and that question is, who are you? And if I would introduce myself, I could say, who am I? I would say that depends on what you analyze, right? So you could say, you could define me based on, or I could define myself, I suppose, based on where I come from, a small town in Utah. Or you could define me based on what I look like. I'm, a 5'10" white male. Many scientists define me what I do, and you could define me by how I'm a scientist or engineer. You could define me based on where my ancestry came from several hundred years ago, or you could define me based on, I'm tired or wanting to play basketball. There's many different things. It depends on that data that you choose to define yourself. The other thing is how you analyze that data. So, if I quantified all the hours I spent doing different things, you could see that you'd probably call me a sleeper because I spend most of my time sleeping. If you looked at something like how I'm changing over time, something dynamic, you could see I'm slowly changing away from chemical engineering towards a systems immunologist engineer. If you looked at all the different traits about me and compared them to everyone else, you might find that the top different things is that I like to collaborate and do science and focus on building new technology, and I like computational problems, and I really enjoy teaching and mentoring. Or you just might find out I really like chocolate. Um, I say these things because as scientists, we have our specific views of what a cell is. And how we define a cell is very important to the studies that we do and the technologies we build to understand more about what a cell is. And I'd ask you another question. How would you define yourself if you were a cell? And um, so, following on Yuri's talk about AI, we can also ask AI to define what a cell is if it was a cell itself. And I'm not going to read this, but it's kind of funny that it picks certain things. It's true what it says, but it also has a certain angle, and it maybe leaves out some important things of what our individual cells do. And I'm not saying one way is the specific way to look at a cell, but I will share the way that we look at cells. So our bodies are amazing things, and it's really fun to be at the frontiers of studying how our bodies work and function. And how we see our cells is if we look at any one organ, we can take the intestine, the lung, the heart, any of these, even a diseased organ like cancer, if we zoom in, we'll see these multiple functional, what we call tissue units. The organs, there's different parts of your organs that are all located together, and the different functional units have different functions one to another. And these are subcomposed of specific cell, what we call neighborhoods. Just like we live in specific neighborhoods within larger cities, these little cells are all organized, and there's different compositions or amounts of these different cell types within these. And then within these little cell neighborhoods, each individual cell is interacting with another cell, and these can be different types. It can be immune cells, they can be blood vessel cells, and they're all signaling to each other with these small molecules, both on the cells and between each other. And then even within a cell, there are all these chemical reactions that are going on that's determining what the cell is doing and where it's going. And it's, we can see this across different scales, going from the molecular all the way up to our organs. And what science has done up until now is, we've looked at things in isolation largely. And we've looked at things at the cellular level or at the molecular level, even the organ level. But what we haven't done so much of is look at things across all these levels and connect them and analyze them across all these levels at once. And partially that's been due to a technological limitation. Just our techniques are really good at individual scales. And that's the same with our computational approaches up until now. I think of this kind of in a geographical sense. If we think about our bodies being analogous to the Earth, we can see these different scales happening as well. We can go to a city, we can go to individual neighborhoods, we can go to individual teams, interactions of people, and then we can go to the individual. And that's a kind of similar analogy to cell neighborhoods that I see. So it's, how do we measure this now? And so there's been some really great development in the technologies we use and acquire this large data. And a lot of this uses microscopes and robotics, and the unique pairing of the two in order to do this. So there's many different types of microscopes. Here's an example of one that we have in the lab. And these are microscopes that are powerful enough to see our cells and also paired with our computers that and allow it to be automatic and be able to do things and image terabytes of data automatically without us having to be there and take every picture. We can connect this with fluidics that also allows us to do many experiments at the same time and many different steps and reagents that allow us to take many pictures and look at many different things about our cells at once. And so we can combine these robotic fluidics to these microscopes to do really cool things. So one of the things that technologies that we built was a multiplex, what we call multiplex imaging, which allows us to basically measure 50 different molecules at once at individual cell scale. We can use that to define individual cell types and where they are in the tissue, and then use additional computational tools to determine this multicellular structure, and finally look at these network interactions across different scales. And, we spent a lot of time developing different protocols for this, thinking about how you process all this data and store it, the type of metadata that you have to take along with this to make sure that it can be standardized and analyzed across other experiments later. How do you develop these molecular tools and do it in a way that's not too cost prohibitive? How do you think about standardizing the data analysis and actually making sure that your measurements are going towards the cell types? And then how do you think about ontologies? And a lot of these didn't necessarily produce the data that we're actually using for biological insights, but were really important steps in order for us to be able to trust the insights that we have. So this is an example of one of these images from this technology. And basically what we're doing is we take a tissue. Let's say a patient goes into the doctor's office and gets a biopsy. That biopsy gets cut into really small, thin sections, put on a slide, and then we put on these molecular paints, which are basically DNA conjugated to antibodies. And then we read them off and visualize them with our microscope. And each individual color represents a different molecular paint that's targeting a different molecule, and we can see that our individual cells here are expressing different molecules. And this is a piece of the healthy human intestine. Here is the lumen of the intestine. So over here would be where the food is getting digested. And this is the barrier, which is called the mucosa. And underneath it, there's some stromal area where you have immune cells, lots of vasculature, which you can see in yellow, and then you have the muscle area of the intestine. And so this is a cross section of the intestine, and we're imaging it with these molecular paints. And we can do many different types of paints. Here's a zoomed in area. And we can look at many different markers at once all across the intestine. And each of these help us learn about different areas of the intestine, different cell types that are all working together to do something. And all of this is happening at the single cell level. So if you see these individual little circles, this is an individual cell. Here, these are immune cells all working together in what is called an immune follicle. And then here you can see these long cells all next to each other. These are endothelial cells, which are vasculature. And you can see this big hole, which is a blood vessel. And that's where your blood vessels are transporting nutrients. And so, we as humans can see there's natural structure and we can identify different structures. And that's what we've done for a long time. But, beyond pretty pictures, we want to make this something that's more automated because of the terabytes of data that we're generating. So what we do is we apply machine learning algorithms or AI to help with this. And we do this in many different ways. One way is in segmenting each of these individual cells. We can apply AI algorithms that do this for us. Basically, they draw a little circle around each individual cell. And then, like Jure mentioned, we can develop AI algorithms also to help us identify and label the different cell types. So for instance, these magenta cells that were in the mucosa, we know these are epithelial cells, or the cells that maintain the lining or barrier between our intestine, the outside world, and the inside world. And what these algorithms can do is, you can see here's a ground truth of what the cell types are. Each individual dot is a cell within the intestine. And then if we leave this out and we train on this data and then predict, we can see we get good concordance with our predictions. So AI, it plays a big role in helping us go from a pretty picture to quantified information that we can do something with. So how do we identify these larger cellular neighborhoods? One of the ways that we can do that is by thinking about these cells as neighborhoods. And one of the computational ways we do this is to look at a window of cells, count up all the different cell types in that window, and then we put those into different groupings. And ones with similar compositions will group together into cell neighborhoods that we call, for example, these two are more similar in composition than this one. And we can name these based on the cell types that are in them. So for example, here is our Codex imaging data, this multiplex technology. Then we derive our cell types using these algorithms I talked about before. And then if we do this neighborhood analysis and try and group different compositions, we can start to see these structures. Here's this inner and outer follicle, which is this immune region. We can see vasculature in yellow throughout the intestine. And, this is a quantitative approach. And so we can quantitatively see which cell types are depleted, which are in blue, or which cell types are enriched, which are in red, within each individual cell neighborhood. So these are the individuals that are mostly present. So for example, in this neighborhood, we have plasma cells, and CD4 cells, dendritic cells, macrophages, and is a neighborhood that we call the plasma cell enriched neighborhood. And, we can identify these different neighborhoods, but how can we analyze across many domains? Because, you can think about this vasculature is very different from this vasculature, that's very different from this vasculature. And kind of like what I mentioned before, is we already have a sense for this geographically. You can think about neighborhoods within a county. And then we can think about counties that then make up a state, and many states that then make up a country. And so we did this for this healthy human intestine data set, where we identify first all the individual cells in many different sections of the healthy human intestine. After we did that, we used those to identify neighborhoods across the intestine. You can start to see organization of different areas. As you move up to the intestine, we can see which cell types are enriched in which neighborhoods and start to build these complex graphs. And then we can do something that we call communities or neighborhoods of neighborhoods, which is a higher spatial domain, where we start to see even more structure as we analyze the data this way. And then finally, we can go even further and do neighborhoods of communities that are these larger tissue units that come together to make up a tissue. And we can see how everything is connected across from molecular all the way up to tissue level. And these connections can tell us a lot about what's going on in the tissue. I won't go into too many details, but one example was one community called an adaptive immune-rich community. We saw this was the intersection of multiple immune cell neighborhoods and epithelial cell neighborhoods. And it was actually an important finding that we found that these immune cells, particularly T cells, were enriched towards the bottom of our mucosa, or this barrier between the lumina of the intestine and other parts of the intestine, and found that these were enriched in the stem cell zone. So it's findings like this that can help us understand more about how our organs are organized at the cellular level and how that contributes to their overall function. Another example is I've been a part of another project where we're trying to understand why chronic inflammation can lead to cancer. And it follows a shared pattern where we're going from a normal barrier in many cases, like in the esophagus, colon, stomach, or lung, to, and there's special cells called our epithelial cells that create this barrier. But when they get injured, and especially when they're chronically injured, there's a chronic inflammation, which means there's a lot of immune cells there. And when this happens, a certain percentage of patients will develop what's called metaplasia, where their epithelial cells transform to a different type. And then some of these patients then go on to develop what's called dysplasia, which becomes more aggressive. These cells divide much more frequently and don't look very healthy. And then a good portion of these patients go on to develop cancer. And so it's not entirely clear why this happens and why it's associated with chronic inflammation, but we assume that this chronic inflammation has an impact on the development of the cancer. So we applied our technology to these samples from patients. And we analyzed them in the same way, where we're looking at these cellular neighborhoods. And one of the different types of analysis that we applied is something that we borrowed from ecology, is to look at how diverse our ecosystems are. And one of these metrics is called a Shannon's diversity metric, and that just measures how diverse an ecosystem is, and that's based on the number of species within an ecosystem and the percentage representation from each species in an ecosystem. So we took this principle and applied it to our Codex neighborhood data. And what was really interesting is that in the normal type of epithelial, it had the least diversity, whereas in the cancerous part or neighborhood, it had the greatest diversity. And this is really interesting because what we found, what we know from the metaplasia is that it goes from a normal epithelial to metaplasia, kind of like the intestine, so this is from the esophagus now, but it shouldn't be like the intestine. And then it becomes more aggressive, and then it de-differentiates to more like a stem cell-like state. It doesn't look like the esophagus or the intestine. But what we found is that the stroma, or the area around it, also is doing the same thing. So it goes more towards like environments that we saw in the intestine when we applied this data. And then it de-differentiated or became more diverse in the number of cell types, especially immune cells, as we continued to study the disease. So this indicates that the stromal cell types could be a very important therapeutic target down the road, where we could develop therapies not just to the cancer, but potentially to the stroma, to reverse this phenotype or de-differentiation of the organ there. And speaking about therapies, another type of therapy are immune cell therapies. And basically, what these therapies are is that we can take out our immune cells, we can manipulate them in genetic ways or with chemicals, and there are a certain number of cells that can recognize cancer, for instance. And there's other diseases where this is used, but primarily where we're interested is using these for cancer. And these immune cell therapies are very useful because they're living, they can be transferred back into the patient after you've activated them or grown them out to be large armies of immune cell therapies. And they go back into the patient, and they can traffic or go through the bloodstream until they find the cancer, and then they can go and kill it repeatedly many, many times. But, you know, and this comes into a thought, you know, in biology, you know, we can think about our neighborhoods. I've kind of talked about these neighborhoods as like static things, kind of like geography, but really our cells are dynamic things, and there's different types of teams that are working together, kind of like a company. So, you know, we can think about an engineering part of a company, a sales part of a company, and like an R&D part of a company. And so, you know, trying to figure out what different cells are doing and how they're working together is another way of thinking about our tissues at this scale. And what we found is that there are teams of cells and they're working together to do specific things. And when we were studying these tumors in patients treated with immunotherapies, we saw that there were specific teams that helped patients do better. And these were not present in patients who did not do as good. And some of these were, so if you look at this tumor, there's a lot of complex data here, but specifically, hopefully you can see this light blue area next to this dark blue area. And this light blue area was a team of basically lots and lots of different types of immune cells. And it was when this team was right next to this blue team where was our T cells actually going in and killing the tumor, when those two were next to each other and present in large amounts, we saw better outcomes for the patients. And this was, using our technology to really see both biology within human patients and also some new finding that we can base further development of these therapies and also potential stratification of these therapies for patients is really exciting. So, one other thing, hopefully I can get into before I go to questions is, I've talked a lot about deconstructing complexity by these awesome technologies, but as an engineer, I'm also interested in building it. And so one of the ways that we can build it is computationally. And we can do this by creating computer models of our tissues. And one way we can do that is by giving cells to be agents, and we can give them rules and properties. We can tell them to interact with other agents in the simulation environment. We can put them in a spatial environment, and then we can get emergent properties that happen. And the most classical example of agent-based modeling is ants. So in this example, the ants only have a few simple rules. Walk around randomly until you find food. When you find food, put down pheromones and go back to base. And then if you find pheromones, follow the trail until you find food. And by just those simple rules, you can get this very, similar behavior that you would imagine to ants. So we did this. We created an agent-based model of T cells and the tumor microenvironment because we wanted to learn more about this behavior, and especially dynamically. The images that we take with our technology currently are just static images. And so we can start to study things of how does the tumor develop over time? When we add T cells, how do they go in and kill the tumor? And if we change the type of molecules that they express, can they kill the tumor better? And this really allows us to understand what's going on in these microenvironments and how cells are working together in teams and where and what abundances are really important. And so, I'll just leave with a closing thought that, it's really important every time we develop some new technology, we are able to discover some new biology. And then based on that biology, that leads to either a new diagnostic or therapeutic that helps human health. And hopefully with these multiplex technologies, right now we're just in the biology discovery phase. And hopefully we can take that and use it to create new diagnostics and therapies that are more holistic thinking about these interactions from the molecular scale all the way to tissue scale. And that we can start to create some of these complex multicellular structures or neighborhoods or destroy them if they're not good. And with that, I'll just say that my lab here is exploring these things at Duke University. We're really excited. I have a group of really amazing people that are part of this, that are across many different disciplines. And we are happy to take questions and yeah, thanks again for the opportunity.
Thank you, John, for an excellent talk as always. If there's any questions, please put them in the question and answer box that you'll find at the bottom of your Zoom. And then we can go ahead and ask, or I think if you raise your hand, we might be able to unmute you. I don't know. Is that true?
Hi, John. Really great work. And I love to see the agent-based models. That's exactly what we need and what we can also visualize ultimately, right? So I put in the Slack that you actually inspired you and others, the Squiggy's identity crisis scrolly story. I don't know if you saw it, but it's actually quite funny. So it's a cell and it doesn't really know what it's called. And then it's trying to figure out what it is and then has a little companion, which is helpful. And it's totally nerve-wracked about what am I, right? And so you started your presentation this way, and this is really meant for general audiences. So I appreciate that. Going forward, I know you're also working with Ushma on another video, and I was hoping it would be done, but I think Ushma is still working on it, and hopefully next time we get to see it. So thank you so much for also doing outreach. I wonder how students that get excited about your work would connect with you, would become your students. Probably you're busy with the students you already have, but maybe there are opportunities for students to attend summer workshops or MOOCs, or what else could they do to get closer to your work?
Yeah, great question. Well, this is something that's always developing. I feel like we're finding newer and more creative ways to get people involved. We're hosting our first ever hackathon this next week, but we're doing it at a small scale, the first one, and hopefully we'll continue to grow things like this and become more outward facing and integrating many people. Um, we're very excited about other outreach type things and citizen science type of research and using tools that don't cost too much, that don't require university level funding and can get a broader audience involved. I think we're very excited about these type of things. Right now, probably people like just using my email from Duke or wherever you guys have posted on this event is the easiest way to contact me. But yeah, in the future, hopefully we'll continue to follow in your footsteps, Kadi, to engage with people and the community.
Yeah, thanks for being here, John.
Can I ask one science question? For your chronic inflammation associated cancer studies, have you looked at all on the microbiome scale and whether the microbiome plays any role in that situation of inflammation?
We didn't unfortunately do that. Out of the many assays that we did not do that, but it undoubtedly does. Particularly, I mean, there's research to show that it does,
Yep.
but the other thing that was really interesting is that one of the new cell neighborhoods that showed up in those um, tissues as they transitioned was this one called plasma cell neighborhood, which is the example that I showed in the intestine. And the plasma cells are the types that we see are specifically types that are generating antibodies against microbiota.
Mm.
And so yes, they're really important, but we didn't explore them too much in our study.
Okay, thank you.
Yeah.
All right, well, I think we're at the top of the hour, and I believe that this ends our session, and Andy's session comes next. So thank you very much, and always exciting to hear about your research. Thank you, John.
Thanks, Ellen. Really appreciate it.